staining of bacteria is also called Gram staining. In microbiology and bacteriology, Gram stain is a method of staining used to classify bacterial species into two large groups. The two large groups are gram-positive bacteria and gram-negative bacteria. The names comes from the Danish bacteriologist Hans Christian Gram, who developed the technique in 1884. Basically, Hans Christian Gram did one experiment, identification of bacteria, mainly two bacteria. One is gram-positive bacteria and gram-negative bacteria. See, during this procedure, we are used bacterial smear, and the smear is taken with inoculation loop on clean slide, and the bacterial culture make a smear, a thin layer appeared on the glass side, and it exposed to air called air drying. After air drying, after bacterial smear preparation, it stained with crystal violet or gentian violet for 30 seconds. Later, the culture is turned into a bacteria get stained with bluish purple. When we added crystal violet or gentian violet to the bacterial smear, it turned into bluish purple. Later, the smear, a bluish purple bacterial smear washed with water and iodine staining. The iodine staining containing 0.5% solution in iodine. That type of iodine used for the staining purpose in bacterial staining. Iodine works as a moderate, and formation of crystal violet iodine complex, CVI complex. See, initially we add or we stain crystal violet, later wash with water, again stain with the iodine. So crystal violet iodine make a complex on the smear. The smear complex is also called crystal violet complex, crystal violet iodine complex. Then later it treat with alcohol. Some bacteria retain the stain called Gram staining. Retain the stain means they are always in blue color, bluish purple color. So that type of bacteria are called gram-positive bacteria. They containing at the outside of cell wall thick polysaccharide layers, and some bacteria, de-stained bacteria after treatment with alcohol, de-stained bacteria, and the de-stained bacteria counter staining with safranin. The de-stained bacteria get recolor, pink color appearance with safranin. So which bacteria turn into pink color? That bacteria are called gram-negative bacteria. At outside of the bacterial cell wall, thin polysaccharide layer is present. That bacteria are pink in color. The bacteria are also called gram-negative bacteria. It is the basic procedure of gram staining technique for the identification of gram-positive bacteria and gram-negative bacteria.